In this class, we're going to talk about four main topics. In the beginning, we're going to give a little bit introduction about regeneration during evolution. Then we're going to have an example of the flatworm regeneration. And then we're going to focus on two species to show you that even close species, closely related species, can have different degrees of regeneration. And then we're going to talk about the windless pathway, how a small change in a pathway can can bring back the regeneration that is lost during evolution. Okay, so let's start now. So let's start with the regeneration, and during development we can have some degrees of regeneration of some part of our bodies. However, we start losing that degree of regeneration during adulthood, and the older we get, the less likely that we regenerate cells. However, all organisms, they can regenerate those structures during not only development, but during their all lifetime, and we're going to talk about some of those extreme cases. So I know that maybe you have here examples of beautiful animals like these ones that have examples of regeneration. Yeah, maybe the flatworm, a beautiful example of regeneration. Some of you maybe in some of the courses you already have done those cats to see the flatworm regeneration, or maybe it's more difficult to think about starfish, how they regenerate an entire arm. And in humans, we can regenerate to some degree part of our liver. But the question is like why we can, even humans can regenerate part of our liver, but not part of our eyes or our adult hearts. Even the coronary arteries, they cannot be regenerated. And we're going to talk about these kind of things. This is a suggested literature. You don't have to read it only if you're very passionate about these things. One day, if you have time, there is a lot of information about regeneration. This is already relatively old. There is even more and more articles about regeneration. So here we have an example. Newts are able to regenerate the limb and the jaws and the lenses. Yeah, so it's very interesting that they have a high degree of regeneration for a vertebrate. Another example is zebrafish that is able to regenerate the fin and the heart. And yes, zebrafish is a model organism. We can study how they can do it. Deers are able to regenerate the antlers so they grow every season and they develop again. Flatworms are able to regenerate the body, and we are going to see some example of what type of cuts can be done and in which species. And sea stars are able to regenerate the intestine and the arms that are very famous. And Nigerians are also an example, a beautiful example that they are able to regenerate the body. The sponge are also able to regenerate the entire body. Yeah, you can break it and you can put them break it into small pieces, and they can go back and regenerate. And the question is why some of them can do it and why one cannot do it, and why during evolution some of them they still can, like you need to be a complex organism. You can be a complex organism and still regenerate some of these structures. So the idea is to study the molecular basis of that. And in this class, we're going to go again and give you a very general example of something that is happening there during development. So this is a diagram that I took from this article, and here we're going to talk about the changes in the regenerative ability through phylogeny, ontology, and age. Okay, so here we have the development, the adulthood when they are young, and when they are old. And here, for example, we were talking about the antlers, but they can, so that is bone, cartilage, and skin. So they are able to regenerate the antlers, but not an entire leg. So it's fascinating that they can do those kind of things. Here, mice and humans, they are able to regenerate the same degree, like it's very similar where we're able to regenerate. Uh, and and here the two examples of flatworms and Nigerians are just amazing. And I'm going to show you a video. Maybe if you're watching this class online, I'm going to just show you the link that you can go. I'll post it in Populi or in Moodle, so you can give it a look there. So in case that it cannot be added, I cannot add it in YouTube here. So you can watch it, but it's very interesting examples of organisms during evolution that are able to have different levels of regeneration. And here in this phylogenetic tree, you can see in red some of them that are that high levels of regeneration, while in blue some of them that have low levels of regeneration, and the losses of the potential for regeneration are here with a cross. So for example, here you see that in many cases fishes, amphibians, reptilia, and birds and mammalia, they lost the ability to regenerate. Well, same thing happened with arthropoda, but well, look, Nigerian porifera, yeah, sponges are able to, they still keep it. And if you go in detail, some of the amphibians of the reptilia, they can also regenerate some things, some parts of the body, but it's just fascinating. And so what I'm going to show you is that if you go in detail and study even these ones, yeah, like the flatworms in detail, you will see that that some closely relative are not able to display regeneration, but you can bring back the regeneration by studying those pathways, the signals that create these uh, these mechanisms, so these molecular mechanisms. Okay, so we talk about regeneration in terms of phylogeny. Let's now talk about in terms of types. Yeah. So first type is the during embryogenesis. So we talk about this that if, for example, in frogs, you are able to regenerate. If you kill one of the cells, you are able to in the early embryogenesis, you are able to regenerate it. Yeah. Something similar happened during organogenesis. Yeah, and for example, in flies, you can remove uh, through dissections pieces of the pupil, yeah, the legs, and they are able to some degree to regenerate the entire leg, or they can are able to just produce a fly that is just a little bit smaller, but is able to if depending on the change on the damage, they are able to do that. So okay, so during embryogenesis it can be done. During larva or fetal, they can also be done. Yeah. Um, Also during juvenile, there is some degree of regeneration that we talk about. Even kids that in early stages of their life, the tips of the finger tips and parts of the fingers can be regenerated. And although this ability is starting getting lost, and during adults, there are few examples of high regeneration, like the antler that we have been talking. Okay, so it seems that the older that you get, the less regeneration that you have. Yeah, but there are different type of regeneration. And here I show you just the diagrams I told you about during to remove part of this embryo. So if you take an part of the embryo and cut those cells, they will be able to to fix the damage and produce a normal organism in many cases. In some cases, if the damage is too big, they will be able to produce a smaller organism. Something similar happened with winged legs. You can, in the early stages of the development, cut a piece of the wing or a piece of the leg and regenerate the structure. In humans, in infants, I told you about the finger. Pieces of the finger can be removed in the distal phalange. Yeah, we don't know how this happened, but they still can do it. A much axolotls, they can do that much better, and we still don't know all the details, but there is a lot of research in that. And the king of all of this is the planarian, that this is something that we're going to study today. Okay, there are different types of regeneration. So one that it has been studied in detail is the one that is mediated by stem cells. And this is what happened, for example, in the skin, muscle, guts, and blood cells. So all of us, we have stem cells. And when the cells die, they can differentiate and create more of the old cells. Yeah, and so we can definitely still do that. This is how our skin still reproduce. Yeah, so basically in a tissue you are going to have these stem cells, and on the top you're going to have all cells that are dead in the skin. The other type is the compensatory regeneration. So for example, what happened in the liver, that you just create this mitosis that produce those type of cells. If you take genetics, and you will see that stem cells have wonderful genetics, that it allowed them to correct their DNA. So it's good, but at the same time bad, because to be able to correct your telomeres, for example, the length of your telomeres, it could be good, but it's a characteristic, for example, of cancer cells, that they are able to not respond to apoptosis, to dismiss some of those signals. So anyway, so that is fascinating, and sometimes I wonder why not all the same have the same potential, stem cell potential, and this is possibly one of the reasons. Okay, and this is one of the videos that I think it will be very nice that if you watch it, it will cover more models, organisms for regeneration, and it can explain more things in a fun way and interesting way. I cannot put it in this YouTube video, but I will put it on Moodle or on Populi. Okay. Okay, I just wanted to finish this first section reading this nice quote from the book. So regeneration is the reactivation of development in the post-embryonic life to restore missing and damaged tissues. And it's pretty cool. This is exactly what happened, for example, with our skin. This is why we're constantly renewing our skin, because the development is reactivate and it keeps happening. It's a shame that we cannot do it with other parts of our body. Okay, we just finished with the introduction of the regeneration. Now we're going to talk about the flatworm and the regeneration. So let's start. Okay, so maybe you have been able to do this type of cuts, and you can imagine this. What happened if you cut this put flatworm half and half? Yeah. What about if you cut here and here? What about if you cut it laterally? Yeah. Yeah, so there are many ways in which you can cut a flatworm, and the question is in which one they will regenerate. Yeah? So if you cut it the intermediate section and you put this one two together, if you cut the head and the tail and you keep just the middle part, and what about if you cut the lateral section? Yeah, the left or the right. So what are the things that you're going to observe? So in some species, you will observe this, that they will develop and regrow the part that they miss. In some species, they can also regrow the head and the tail. and in other ones they can regrow the left or the right part, and it's pretty amazing. And if you have done those experiments, I think it's pretty cool to see it. So now that we know that they are able to regenerate, so let's talk about the in more scientifically what is happening. So the terminal regeneration, the one that we saw with the red line, is missing the head. The green one is missing the tail. So it's pretty incredible that the organism is able to knows what part is missing. Yeah, if it's missing the head or is missing the tail. So this is something that it still is able to know. And we are looking for the molecular mechanisms that they are able to know this polarity. What is head and what is tail? The other pretty cool question that we have is how they're able to recognize that we have a left. Now we keep the left, now we need the right. How they are able to keep this bilateral, this um, the lateral half. And we, in some cases, there's some researchers that study this type of questions, like why we have one eye and we have two eyes, and how the body is able to know if they have the same size. Yeah, and in this case, with this type of research, we are also doing something similar. Is like how they're able to know not only that they are missing the right part, but that they are able to accomplish the to regenerate the same size again, and they're able to follow the same functions. Okay, so this is just pretty cool questions to pretty cool experiments. And finally, the intercalary regeneration, that is also fascinating, how this organism is able to uh, put this part together and able to say there is another piece, the middle piece is missing, and we are going to work together. Yeah, although we are different parts to bring back the piece that is missing. Yeah, so this is fascinating, and we're going to, at the end of the class, hopefully investigate, like you will talk about some of the key experiments and some of the key pathways that they have been studying. Okay, so let's start talking about the difference between planary, the flatworms, and humans. The flatworms only have 10,000 cells. That is, it sounds like a lot of cells compared, for example, with C. elegans, that is much less. And humans have 37 trillion cells. And here they have 30 different cell types. And here they have more, human, we have more than 200 type of cell types. So we don't know if to be such a complex organism, this is the price. The lack of regeneration is the price for the complexity. But this is a hypothesis. Maybe in the future we will be able to regenerate more, parts of our body. So we will see. The flatworm, here you can see the anatomy. So here if you have seen it, the work with the eye spot are here on the head. Here you find the auricula. The pharynx is here, the vascular cavity, and they are just very simple. Here is the brain, and all the nerves are located here. So big picture, not that I want that you memorize everything, all these things for the exam, just that you know that they have structures that are very simple, but they are definitely simpler than humans. But they have regenerated all these structures. Seems to be at this point for humans, it's still impossible to regenerate a simple structure like this, as far as I know. So here you can see, maybe if you have done the experiments, here is what happened when you do these regeneration experiments. You go into asexual reproduction, and this head regeneration will take around 7 days. And the day 0 will be the amputation, and then the wound healing start. Later we will see that there is then the cells that are lost require proliferation. So you form something that is like a wound that is called blastema. That is the wound, the repairing wound. And then after all the cell division, you go into differentiation to produce again the head. So here I'm showing you what happened, for example, for the regeneration of the axle of the arm, that in some way mimic, recapitulate some of the same process. So first it's an injury, then the cell migrate to the amputation plane. So many cells are going to migrate to the amputation plane. Then there will be a wounded epidermis. Later, we require the specialized wound epidermis. It goes to apical epithelial cup. Then you produce it here, and then you form a blastema. And in the blastema, what is a blastema? A blastema is a heterogeneous cell mass that through migration and proliferation is going to form an injury site that undergoes morphogenesis to form the missing organ. Okay, so you see to some degree it's similar to what happened with uh, with the flatworms. You have an injury, you have to cure it. After you seal it, you need to start rebuilding the wound. And how you do that? With the same process that you start in the beginning: cell division, cell migration, and differentiation. In the beginning of the semester, we talk about morphogenesis in terms of organogenesis, but here we're talking in terms of the adult form, because yes, here is a new process of organogenesis, as we read in the textbook, that is the reactivation of morphogenesis during the adult stage. Okay, so here you have a diagram and a picture. Here you see a picture taken from your textbook, and here you see some of those cells that with two staining DAPI for the nucleus and the phosphohistone that is labeling for the histone, that is the places when there is mitosis, yeah. And the phosphohistone is of course the histones. So you see that here in many places there is cell proliferation. And here we have these cells called neoblasts that are, and here in this diagram what they are showing you is that there are neoblasts everywhere scattered through the worm. Depending on the flatworm, they could be even up to 30% of the cells could be this type of neoblasts that are just stem cells that are pluripotent, that are ready to repair any of this damage. And so in this slide, I want to show you that the neoblasts produce the cells of the blastema. So they are going to be the ones, the new cells these stem cells are going to go to the injury, to the injury site after it's sealed, to create proliferation, migration, and start a new morphogenesis. So let's see the steps. So here you see that there was a cut here, and the neoblast migrate to the wound site. The first thing that happened here in purple, what we are showing you, is that there is apoptotic cells at the amputation site. So the cells that are not working well, they died. Then there's migration of the neoblasts to the wound site, and if you see they start changing in color, these represent the different type of cells that are differentiated. Let me show you that. And this, so in blue you see the stem cells, in purple it was the apoptotic cells, and this is true, the pathway that is here, the Wnt pathway, that you have to read it, and we are going to talk more in detail in the following slide. So let me show you more. So what happened here is that in green you find that the neoblasts start dividing, yeah, and form the blastema that is located here. And here you see that in red is the late division progeny that is start already and probably start going through differentiation at the end. And if you see, it not only happened in one side, it happened in both sides. Okay, now we're going to focus on these two species of flatworm and how different is their regenerative potential. So let's start. Okay, so now let's start discussing this diagram, this diagram and this figure that are taken from this article. So here you see that here there was the first dissection, and the first dissection they cut it here, they removed this part, and look what happened. That was the first part that regenerate. Yeah, yeah. Time post amputation, they regenerate fast because it was the fragments with high total percentage. Yeah, so this one was that regenerated the fastest. And now, and they keep doing that with all the rest. Yeah, so they did that with this one, with this one, with this one, and with this one. And of course, with that one, the purple one, it was the one that it took the longest to produce the entire structure. So the idea is that in this species, they can regenerate heads from tail fragments. Yeah. Now we're going to show you what happened with another species. Okay. So just here I show you that all of them end up in 100%. So you can see it. And here you can see that it's the same type of experiment. However, they are not able to regenerate the heads from the tail fragments. Yeah. So for example, the ones that here that they regenerate, that they remove all this region, they are not able to do that. The blue one also is here, and they cannot do it. The green one is only able to regenerate a little bit. So the only one that is able to regenerate is the first one. The one that is miss only this one. The other ones, they were not able to regenerate the eyes. So it's something fascinating how these relatively close species are not able to lose the regeneration, well, or one is able to keep the regeneration while other one it doesn't have. So what they did is to take that species that don't regenerate the head, and they try to study this beta-catenin function as a head inhibitor. Maybe this is inhibiting the process. Yeah, through an evolution, this molecule was able to inhibit the production of the head. So here they did the experiment. In yellow, you will see the control where there is the normal beta-catenin expression, and here where they use an RNAi of interference that is going to remove the beta-catenin expression. Okay, and let's see what happens. So look what happened in the control when there is a normal beta-catenin expression, the function, the normal signaling, there is no production of the eyes. However, when you start seeing here, when we remove the beta-catenin expression through this genetic tool, we start seeing the regeneration. You remember that we told you that find it, move it, find it, lose it, and move it. So here is one of those type of experiments. So after 3 weeks, you can see full regeneration in some organisms. So it's pretty fascinating that only one molecule it was able to allow that this species is able to regenerate again the head. And just in case that you don't understand what is this RNA interference, RNAi. So this one, what we are doing is, this one is, you generally have transcription, transcription, DNA. Here we go to translation. What we're doing is producing an extra RNA that it will be complementary, and they will attach to each other, so they will cancel each other, so there will not be proteins for the translation. So it's a very specific genetic tool for produce genetic perturbation. And in case of the planaria, they just have an injection with this RNAi, and it's very easy to produce. And you put it in the planary, and in that way you remove the normal RNAi, and you are able to produce this genetic perturbation. I hope that this is clear. If not, you can Google RNAi. It's pretty easy to understand what it is. Okay, so we already talked about regeneration. Then we focus on the flatworm as an example for regeneration. Then I show you that the flatworms have molecular mechanisms to study that, and two species that they were different. When we manipulate those pathways, we can start investigating where are the sections where this type of functions are. And now we're going to investigate this Wnt signaling pathway, that is the one that is involved with this beta-catenin. And I know studying pathways sometimes is a little bit difficult, so I'm going to try to show you not many pathways, but this one is just fundamental. And there are 4 different components that you will see every time that you see a pathway in cell biology and developmental biology, cancer biology. There is a receptor always here. Here, later there will be a transduction signal between 2 and 3, and finally there will be a response in force that it can be, for example, you see that this is off, now this is on, so we change the gene expression. So remember, receptor, then there's a transduction of the signal in this one, that are all these molecules, and then there will be a response that it could be multiple things. In this case is a change in gene expression. And all these numbers 7, 6, 10 is multiple molecules that are interacting. They could be many molecules, but there's always going to be these 4 sections. Okay, so let's continue. Okay, so I don't know if you have watched Bob Ross, and that he always said you can do anything that you want to do, and he start painting and do beautiful things and say, oh, happy trees, and then this technique that we learned last time, we can apply it here, and now we use and make a happy lake and happy mountains and happy sky, and it looked like magic. Why I put that? Because the windless signaling pathway is an ancient pathway that is evolutionary conserved and is involved in so many things. It's not only in flatworm, it's in humans too, it's involved in cancer, it's involved in so many things. And is able to regulate crucial aspects of cell fate and determination, cell migration, cell polarity, neural patterning, organogenesis, and sometimes even opposite processes. They can be regulated by the Wnt pathway. Okay, so very important, windless pathway, very important to remember. And why is called windless pathway? Because it has it was found first in Drosophila and in mice. So from the fly that the phenotype was wingless, so it doesn't have wings, and the mouse that it was int one. So they put those 2 together, and this is why it was called wingless. And there are 3 types of wingless pathways that have been characterized: the canonical wingless pathway, the non-canonical, that is for the planar polarity pathway. For example, the bristles, your hairs are involved into this type of wingless pathway, and the non-canonical, that is the wingless calcium pathway. So I'm doing something that I don't like a lot, that is talking about all the pathways. I try to leave that for other videos and give you very general information, but I'm doing an exception here because this is a very important pathway. So this is something that happens generally, that you have a canonical pathway and then you have a non-canonical pathways, and this list can increase over time. Over years of research, there is even more functions, and I'm sure with windless it will continue being like that. Okay, so now I'm going to put a video for you on Moodle on Popular that talk about the windless pathway in a lot of detail. It will take like 3 to 5 minutes to explain this, and you can see all the different steps. In my case, I'm going to make an exercise in the following section of the video, just for you to try to understand the big components of this. Again, I wanted to put the video here in this YouTube video, but maybe YouTube will flag me again and don't allow me to do it. So again, let's start with the exercise. So I'm sure that you already watched the video, and now you know that when you have no wingless and when the wingless is bound, there are two different stages, and you already know that. So when wingless arrive here, there are so many things that happen. This receptor is very important. It's called frizzle, and this frizzle receptor it attached to this one that is LRP56 that that also here this LGR, sorry, that is phosphorylated. This is what these 2P come, and this is what they come here, and there is another machinery of other structures that come here to produce a structural response. Yeah, here you see this APC complex that is here. When the wingless pathway is there, it break it get destroyed, and it is allowed to that one of these complex go to the DNA to a transcription factor, and now the transcription is on. I summarize this really fast. In the video they talk about that. Okay, so the only way to do it is write it down and take some notes. And I'm not going to ask all the steps, but there will be practice questions that they can help you to practice that. Now I'm going to just do an exercise for you to hopefully make this easier to understand. Okay, so hopefully this is easy. Number one, this FZD, yeah, stand for a membrane receptor that is located here. Another one is DVL that is located here, or you see it here. It's a protein that is a cytosolic protein, so it's not in the membrane, and it will be located here. And this LRP, if you see, is what type of receptor? It's a membrane receptor, it's a cytosolic receptor, or you know that it's it could be helping other receptors. So it's that, if you remember, I already gave you part of the answer. It's helping our receptor, and how those receptors that help other receptors are called. Okay, frizzle is a membrane receptor. The shevel is the cytosolic protein, and RP is a co-receptor because it's helping the other ones to hold the Wnt binding element. So hopefully this was clear. Let's go with another exercise. Okay, so here you have again, you just have to fill the blank for these ones. Hopefully it's not too complicated, and in the next slide I will show you the answers. Please, if you want to stop the video, and then I will give you the answers in the next one. So when there is no Wnt, yeah, there is not the signal, frizzle is inactive, yeah, and when Wnt is here, frizzle is active, yeah. So the Wnt signaling begins when Wnt proteins begin to the extracellular domain of the frizzle. Then the activation of the cytosolic protein dishevel induces this dissociation of the destruction complex, and the beta-catenin is no longer modified by ubiquitin, and beta-catenin is not destroyed anymore. So here is the experiment of the beta-catenin that we were talking last time. Here there is another exercise, and here do you remember how is this called and which are the components? Pretty easy. just color coded, and what is the function of the of this destruction complex, and they transfer a short ubiquitin peptide to do what? Okay, so you can stop the video or watch again the video last time, the other one, and I will show you the answer. Okay, so the destruction complex that you can find here that have all these 5 proteins, including and that the destruction complex is able to bind to the beta catein, and it transfers a short ubiquitin peptide to the beta catein to be destroyed in the proteasome. Okay, and now the how is called this complex here, and what is the function, and I'm going to show you the answer. So this is called proteasome. Here it is, and you already watched it in the video. And this complex of proteases that destroy the beta catein. The function of this beta catein ubiquinated is bound and destroyed by the complex of proteases that are also called proteasome. And this is information that you can find in the video. The last exercise of fill in blank is this one, and is related to the gene expression. So in one of them they have low levels of beta catein, and in our one they might have higher levels of beta catein. So you have to know which one it is, although it's pretty easy, and you have to try to fill the blanks here. So at low levels you have this molecule called Groucho that is going to not allow the beta catein to attach here, while at high levels Groucho is not able to attach, but there's high affinity for beta catein, and this led to the activation of the gene expression, while this one is the inhibition of the gene expression. Okay, so I tried to show you that example of that pathway, and it's a way to remember. Hopefully, I know that is not always easy, but I will show you, I have practice questions for you to study for the test. And now that I show you the canonical, I told you there are other 2. You don't have to memorize it. You just need to know that. I just wanted to show you that some components are still preserved, like frizzle here, yeah. Other ones are completely gone in the other canonical, non-canonical pathways, the planar cell polarity pathway and the calcium pathway. Again, they end up with the changes in the gene expression. And there are things that if you're interested into a master or a PhD, or maybe for a question of the MCAT, they might give you, I don't think that you will need to memorize each one of these steps, but you might need to understand what is a membrane receptor, what is a core receptor, what is a cytosolic protein. Yeah, all these basic things that I tried to give you in this class, in this video lecture. Okay, let's move on to the next section. Here you can see another experiment that can give us a better picture of what is happening here. So again, they did an experiment with a tail amputation, and here you have the 2 examples. One, the normal case, and here when you reduce the amount of beta-catenin to RNAi. And here what happened is that, well, in the normal case, you have the complete tail regeneration. Here you can get regeneration, but you get another head. In other words, it seems that there is a formation of a second head due to the reduction of the beta-catenin expression. Yeah. So it seems that there is a morphogen here. The expression of beta-catenin should be not. There should be no expression in the head, and there should be a high expression in the tail. Yeah, and in that way, the flatworm is able to detect what is the head and what is the tail. Okay, so although the information is much more complicated and there are more details. I think this is enough of that component of cell regeneration. Now I'm going to tell you a little bit more about neoblast. And the question is there only one neoblast type that form all the cell types? And so here you see that what they found is that rather than they found one type of neoblast, they found a many different type of specific type of neoblast that depending on different transcription factors. So here you can see different type of markers. Yeah, here are all the different type of neoblasts. Here the one with sigma, all the one here with zeta, and what they found is that they start organizing the different type of neoblasts and the different part of the body that they will form. So, for example, nu is going to form the brain, zeta is going to form the epidermis, sigma is going to find the muscles, and so on. Okay, at this point, you should be able to know what is regeneration, how is spread during evolution. You should know that one of the examples of regeneration is the flatworm, and you should be able to compare different species, yeah, to different species that they have different potential, and see that the difference in the potential of regeneration is due to the Wnt signaling pathway. And with this, I know that is very brief, and there is a lot of things, there is a lot of reading that you can do if you're interested in this topic. But I think for today is enough. If you have any question, just let me know and I will try to answer it. Have a good day and bye.